So now you can apply acetic acid to the cervix, right? So this theory is based on the fact that in the cervix, we all know that there are going to be normal cells being present in the epithelium, right? The ectocervix contains the squamous cells, the endocervix contains the columnar cells. Okay, so these are the normal cells. If there is going to be any dysplasia happening in these cells, that it is going to undergo malignant transformation. So this dysplasia means there is going to be abnormal growth and proliferation of these cells, right? If there is going to occur abnormal growth and proliferation, definitely the nuclear cytoplasmic ratio is going to get altered. So based on this fact that if you are going to apply around 3 to 5% of acetic acid to the cervix, if you're going to apply 3 to 5% of acetic acid to the cervix, then what is going to happen is that the normal cells will stain pink in color, whereas the dysplastic cells are going to appear white in color. And this is because of the fact that once you apply this acetic acid to the dysplastic cells, in the dysplastic cells, the nuclear protein will get coagulated. The nuclear protein will get coagulated. So since the nuclear protein gets coagulated, then the dysplastic cells are going to stain white in color. So if we are going to visualize the cervix after applying this acetic acid, 3 to 5% acetic acid, right, whatever appears white in color, right, let's say for example this area is white in color and this area is going to be pink in color, this area is pink in color and this area is white in color, right? So from this white area I can take a biopsy now, since these areas signifies that there could be dysplastic cells present in this area. Since dysplastic cells are present there, since the nuclear cytoplasmic ratio is altered, the nuclear proteins get coagulated and as a result of that, this dysplastic cells change white in color. So this is the theory of acetic acid, and this visualization after applying this acetic acid proves to be, the result comes to be that white in color, right? Whereas the normal cells which are not dysplastic, they appear pink in color, whereas the dysplastic cells will appear white in color. So basically you have to take biopsy from anything which is rough, anything which is looking abnormal cervix, right? And then you are not going to take any biopsy from the Nabothian cyst, and you have to take biopsy from any aceto-white areas, right? These white areas are called as aceto-white. Since this white appears after the application of acetic acid, any aceto-white areas you have to take biopsy because it signifies that these are the dysplastic cells. Is that clear? Yes, so as I stated earlier, dysplastic cells will appear aceto-white, whereas the normal cells will appear pink in color. Right? So this is all about the biopsy, colposcopy guided biopsy. Now there is a specialized feature in this colposcopy, and that is all about the green filter. So this colposcopy, we know that there is going to be a focal light coming and the magnification is around 30 times, the focal length is about 30, and this colposcope has a green filter. So through this green filter, you can actually visualize the abnormal blood vessels. So any abnormal blood vessels, if it present in the cervix, right, any abnormal blood vessels present in the cervix, with the help of green filter in the colposcope instrument, you can visualize those abnormal blood vessels through this green filter, and you can take the biopsy from this abnormal blood vessels, right? And these abnormal blood vessels could be punctate blood vessel, mosaic blood vessels, and they could also be reticular blood vessels. So from these abnormal blood vessels, with the help of green filter of the colposcope, you are going to take biopsy from this abnormal blood vessels as well. Is that clear? So this is all about the colposcopy guided biopsy. Now we are jumping into the reports of colposcopy, which is even more important, right? The reports of the colposcopy is even more important. So we stated that the report of pap smear will come as high squamous intraepithelial lesion, low squamous intraepithelial lesion, atypical squamous cells of unknown origin, atypical glandular cells of unknown origin. But unlike the pap smear, the reports of colposcopy is going to be like CIN. What is CIN? C stands for cervical, I stands for intraepithelial, and N stands for neoplasia. So it means cervical intraepithelial neoplasia, and we have divided this into 1, 2 and 3. So what is 1, what is 2, and what is 3? We are going to learn here, right? So CIN1 means that the dysplasia is limited to less than 1/3 of the thickness of the cervix. So let's take a rough estimate of cervix here, right? So this is going to be the cervix, right? So this is the entire thickness of the cervix. CIN1 means dysplasia has occurred in the cervix, and this dysplasia is limited to less than 1/3 of the cervix. So dysplasia less than 1/3. Let me write here dysplasia. So if dysplasia occurs less than 1/3 of the cervix, it is called as CIN1. So this is 1/3, right? And now you see here that the dysplasia is involving 1/3 to 2/3 of the cervix, right? It has involved about 2/3 of the cervix. Since it has involved 1/3 to 2/3 of the entire cervical thickness, it is called as CIN2. So in CIN2, the dysplasia has involved about 1/3 to 2/3 of the cervical thickness. Then what is CIN3? CIN3 means it has involved even more, that is more than equal to 2/3 of the cervical thickness. So dysplasia has involved more than equal to 2/3 of the cervical thickness. So less than 1/3 is CIN1, 1/3 to 2/3 is CIN2, and more than 2/3 is CIN3. Now I have a very important remark here, and that is we have studied that the report of pap smear, if it comes to be low-grade squamous intraepithelial lesion, it corresponds to CIN1 of colposcopy. And we have also learned something called high-grade squamous intraepithelial lesion in pap smear, and that corresponds to CIN2 and CIN3. Is that clear? Okay. Now we have learned about CIN1, 2 and 3. This is 1/3, 1/3 to 2/3, more than 2/3. And we have two other varieties here, and that is cervical carcinoma in situ and invasive cancer of the cervix. Cervical carcinoma in situ, what is in situ? In situ means inside. Invasive means outside. Very simple, okay? In situ means inside, invasive means outside. That's all, okay? So here, when you see the cervix, you can see an overlying intact membrane. As you can see here, there is an overlying intact membrane. And this is the entire thickness of the cervix. If it involves 1/3, it is CIN1, 1/3 to 2/3, it is CIN2, more than 2/3, it is CIN3. Now, what is cervical carcinoma in situ? In situ means that the entire thickness of the cervix has been involved. The entire cervical thickness is involved, and the overlying membrane is intact. As you can see here, the membrane is intact. Membranes are present. So, if the overlying membrane is intact and the dysplasia has involved the entire cervical thickness, it is called as cervical carcinoma in situ. Cervical carcinoma in situ. Then what is invasive? I told that in situ is inside. Invasive is outside. So as you can see here the membranes are not intact. The membranes are actually breached, right? As you can see here, the overlying membrane is actually breached, and the dysplasia has involved even the entire cervical thickness so much that it has even broken the overlying intact membrane. So the membrane is also breached, and the dysplasia is involving the entire cervical thickness with the overlying membrane being breached. So this is called as invasive cancer cervix. So basically, invasive means the overlying membrane is breached. Carcinoma in situ means entire cervical thickness is involved, but the overlying membrane is intact. 1, 2, 3 means 1 is 1/3, 2 is 1/3 to 2/3, 3 is more than 2/3, and all the CIN1, 2, 3, the membranes are intact. So this is all about the cervical intraepithelial neoplasia, which are basically the reports of the colposcopy, right? So these are the reports of colposcopy. Now based on the reports of colposcopy, you can definitely plan the management. And what are the management which can be performed in cases of cervical intraepithelial neoplasia, we'll be discussing in the subsequent modules. I hope as of now it is very clear for you whatever we have discussed, right? Can you please give your attention here? As you can see here, this is the entire cervical thickness, and this is normal. As you can see here 1/3 is involved. Let me use another pen. 1/3 is involved here. 1/3 to 2/3 is involved, that is CN2. More than 2/3 is involved, that is CIN3. And this is invasive, where the membranes is also breached. Sure? Okay. So this is all about the colposcopy guided biopsy, which is a diagnostic procedure, and based on this procedure, treatment can be done. So let's meet in the next module where we'll be discussing about the treatment procedure of cervical intraepithelial neoplasia, that is CIN. Okay, until then please keep studying, students. If in case you have any doubt, please write to us in the Mackler handle.